Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Francisella tularensis is a gram-negative coccobacillus, indicating that its morphology lies between that of a spherical coccus and a rod-shaped bacillus. In humans, it is responsible for a zoonotic infection known as Tularemia, commonly referred to as rabbit fever. This bacterium is classified as a Category A bioterrorism agent, representing the highest level of concern for bioterrorism due to its low infectious dose and the significant mortality associated with infection. Francisella tularensis possesses a thin peptidoglycan layer, which prevents it from retaining the crystal violet dye during gram staining. As with other gram-negative bacteria, it instead takes up the safranin dye, resulting in a pink coloration. Francisella tularensis is non-motile, non-spore-forming, and facultative intracellular, meaning it is capable of surviving both outside and within host cells. It is also aerobic, requiring oxygen for survival. Additionally, it is oxidase and urease negative, indicating that it does not produce these enzymes. Francisella tularensis is a fastidious bacterium that requires enriched media for optimal growth. Notably, it has a strong affinity for cysteine and typically takes 47 to 72 hours to grow on cysteine-enriched media, such as cysteine-enriched chocolate agar, BCYE, and SHAB. Cysteine-enriched chocolate agar, named for its brown color, contains cysteine and lysed red blood cells. Despite the name, no chocolate is involved. BCYE, which stands for buffered charcoal yeast extract, consists of activated charcoal, yeast extract, and L-cysteine. On both of these media, Francisella tularensis forms round, gray-white colonies. CHAB, or glucose-cysteine agar, contains thiamine and blood. On this medium, Francisella tularensis produces greenish-white, round, smooth, and mucoid colonies. Francisella tularensis possesses several virulence factors, which act like assault weapons, aiding in its attack on host cells and helping it evade the immune system. First, it is encapsulated, meaning it is surrounded by a polysaccharide layer called a capsule. Beneath the capsule lies an outer membrane made up of lipopolysaccharide. What's particularly interesting is that, in most gram-negative bacteria, lipopolysaccharide binds to a protein on immune cells, such as macrophages, known as toll-like receptor 4, or TLR. 4. This interaction triggers the activation of the innate immune system, leading to the production of pro-inflammatory cytokines and nitric oxide, which help neutralize the invader. However, in Francisella tularensis, the LPS is inactive, meaning that TLR 4 does not bind to it. As a result, the innate immune system is not activated, much like a thief sneaking into a house without setting off the alarm. Next, Francisella tularensis utilizes its type IV pili, which are hair-like extensions on its capsule, to attach to macrophages. Once Francisella tularensis attaches to macrophages, it is ingested and enclosed in a vesicle known as a phagosome. Normally, the phagosome would fuse with the lysosome to form a phagolysosome, where an oxidative burst would occur, releasing harmful free radicals to destroy the invader. However, Francisella tularensis produces an acid phosphatase called a .CPA. Logically prevents the fusion of the phagosome and lysosome, allowing it to evade intracellular destruction. This enables Francisella tularensis to replicate safely within the macrophage. To support its replication, it requires iron, so it produces a siderophore, small, high-affinity, iron-chelating compounds that steal iron from host cells. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Eventually, once Francisella tularensis has replicated sufficiently, the macrophage bursts, releasing the bacteria into the bloodstream and leading to sepsis. From the bloodstream, it can spread to other organs, such as the lymph nodes, where it causes inflammation and pus formation, suppuration. Alternatively, it may spread to the liver, resulting in hepatitis, to the kidneys, causing renal failure, or to the lungs, leading to pneumonia. Francisella tularensis is transmitted through ticks, rabbits, and deer flies, and infection can occur through various routes. Depending on the mode of transmission, tularemia manifests in 6 different forms: ulceroglandular, glandular, oculoglandular, oropharyngeal, pneumonic, and typhoidal. The ulceroglandular and glandular forms of tularemia are transmitted through insect bites, such as those from ticks and flies. The ulceroglandular form causes an infection in the skin and lymph nodes, while the glandular form primarily affects the lymph nodes. The oculoglandular form occurs when the bacteria enter the conjunctiva, which can happen through splashes of infected material into the eyes or by rubbing the eyes with contaminated fingers. The oropharyngeal form is transmitted via the ingestion of contaminated food or water, affecting the mouth and throat. The pneumonic form presents as pneumonia, which can be primary, resulting from the direct inhalation of bacteria into the lungs, or secondary, occurring due to hematogenous spread to the lungs. Finally, the typhoidal form can arise from any entry point and may lead to a systemic febrile illness. Symptoms typically develop after an incubation period of 3 to 6 days, and all forms of tularemia are associated with nonspecific symptoms such as fever, chills, malaise, anorexia, and chest or muscle soreness. Specific symptoms vary depending on the form of the disease. In the ulceroglandular form, there is lymphadenopathy, characterized by enlarged lymph nodes that may be painful to the touch, along with a single red, raised lesion featuring a crater at its center at the site of the bite. In the glandular form, the primary symptom is painful lymphadenopathy without the accompanying lesion. In the oculoglandular form, symptoms include eye pain, increased tearing, photophobia, sensitivity to light, periorbidal edema, swelling around the eyes, periorbidal erythema, redness around the eyes, and regional adenopathy. For the oropharyngeal form, symptoms consist of a sore throat, difficulty swallowing, and cervical lymphadenopathy. The pneumonic form is characterized by a dry cough, difficulty breathing, and chest pain. In contrast, the typhoidal form can range from acute sepsis to a chronic febrile illness, presenting with high fever, chills, weight loss, headache, myalgias, abdominal pain, diarrhea, vomiting, and diffuse abdominal tenderness. Diagnosis of tularemia is primarily based on serologic tests, such as latex agglutination or ELISA, which demonstrate an increase in both IgM and IgG antibodies after 2 weeks of infection. Alternatively, Francisella tularensis can be cultured from biological samples, including blood, lymph node drainage, skin lesion drainage, or sputum. Additionally, there are rapid tests available for detecting Francisella tularensis, such as PCR and the direct fluorescent antibody test, although these are not yet widely accessible for routine testing. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Treatment involves the use of antibiotics, such as aminoglycosides, particularly streptomycin and gentamicin. Alternative options include tetracyclines, fluoroquinolones, and chloramphenicol. Summary of Francisella tularensis. In summary, Francisella tularensis is a gram-negative coccobacillus that causes the zoonotic disease tularemia, which is transmitted by ticks, rabbits, and deer flies. This bacterium is non-motile, non-spore-forming, facultative intracellular, aerobic, and negative for both oxidase and urease. It thrives on cysteine-enriched media, such as cysteine-enriched chocolate agar, BCYE, and Chachab. Its virulence factors include a capsule with type IV pili and lipopolysaccharide, LPS, which facilitate its invasion of host cells. Additionally, it produces the acid phosphatase ACPA, allowing it to evade intracellular destruction, and a siderophore that captures iron from host cells. Tularemia presents in 6 forms: ulceroglandular, glandular, oculoglandular, oropharyngeal, pneumonic, and typhoidal. Diagnosis can be achieved through serological tests like latex agglutination and ELISA, as well as through cultures obtained from blood, lymph node drainage, skin lesion drainage, or sputum. Treatment typically involves antibiotics such as streptomycin and gentamicin. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!